we know that the garden pea that Mendel used has 14 chromosomes. So it has 7 pairs of chromosomes, 7 homologous pairs. And the garden pea turned out to be a model system because each trait was on its own separate chromosome. So on the homologous pair 1, there was the allele for seed shape. And then homologous pair 2, there was seed color. Homologous pair 3, there was flower color. Homologous pair 4, there was pod shape, and so on. So let's take a look at, so we've been looking at just one characteristic through meiosis and how to get the gametes, the alleles, separated into the separate gametes. So if we have a plant, a true breeding plant that has yellow seeds, it has 2 dominant alleles. It's homozygous dominant. And we know that we can only get big Y alleles in the gametes. We know we can only get an allele that codes for the yellow seed coat. So that is a law called the law of segregation. So let's write that down. Law of segregation. Okay, and it means that only one allele for each trait will end up in a gamete. So one allele for each trait will end up in a gamete. So we've got one allele for the yellow seed color in this gamete, and again one allele in this gamete, and one allele in this gamete, and one allele in this gamete for seed color. Okay, so what happens then when we have 2 traits that we're looking at? Then we need 2 alleles in each gamete. So one allele for each trait. We've got 2 traits, so we need to have 2 alleles. So let's take a look at that, see what that looks like. Okay, so what we're looking at right here, we have a plant right here that produces yellow round seeds. So this plant is homozygous dominant for round seeds and homozygous dominant for yellow seeds. That means that all the seeds are going to be yellow and they're going to be round. And then we've got another plant here that we're going to cross with it, which is true breeding. So both of these are true breeding. It's just that now we're looking at 2 traits. So it is pure breeding. It's homozygous recessive for wrinkled seeds and homozygous recessive for green seeds. Okay, so what does that look like? Let's take a look at how it is that we're getting these gametes right here. So remember that these are the gametes from each of these individuals. So this is representing a whole plant. And then one seed from that plant that's going to be crossed from another seed from that plant. We're going to get one gamete right from this plant, and it's going to get crossed during fertilization with a gamete from this plant. Okay, so what do these look like? So let's take a look at parent 1. parent number 1. And let's go ahead and let's take a look at the chromosomes. It really does help me to really see the alleles on these chromosomes. So we've got 2 chromosomes, remember, because we've got 2 traits. So there is a trait, there's an allele right here, and it starts with round. No, that's seed shape. So we're going to start with seed shape. And then there's an allele on this chromosome, or this homologous pair of chromosomes, that codes for seed color. And we have true breeding, so we know that these alleles are dominant for seed shape, so we're going to use the capital R, and they're dominant for seed color, so we're going to use the capital Y. Okay, so let's go to now parent 2. Okay, and let's do our chromosomes again. So we've got chromosome, oh, that's a big one. Okay, that's fine. Okay, and then homologous pairs. And we've got our alleles on here again for seed shape and seed color. But in this case, they are homozygous recessive for wrinkled. So this is a little r. This allele is homozygous recessive, so we're going to use little r, stands for homozygous recessive, and then little y. Okay, so what kinds of gametes can each of these individuals produce? And we're just looking at 2 traits, so we're just looking at 2 of the chromosomes. Okay, so we know we can get a big R in our gametes. In fact, all of our gametes are going to have a big R, and all of our gametes are going to have a chromosome with big Y. So we know that in our Punnett square, remember, we're going to have 4 gametes, but they're all going to be big R, big Y, big R, big Y, big R, big Y, and big R, big Y. So remember that this is going to be a gamete, but now we're going to have 2 chromosomes in there. So it's got to be together. This one and this one are going to both could go into one gamete. Or this one, this chromosome and this chromosome could go into one gamete. But they both have, it doesn't make a difference, they both have big R's. Or this chromosome and this chromosome can go into one gamete, or this chromosome and this chromosome can go into gamete. So you see how there's 4 different possibilities can go into a gamete, but they're all exactly the same. So we can just represent that by big R, big Y. And then the same's going to happen. Oops, oh, I need a pen. Oh, I could do that. So the same thing is going to happen here. Remember that we're going to have these chromosomes need to go into our gametes. We could have this chromosome and this chromosome, that chromosome and that chromosome, that chromosome and that chromosome, but they're all still going to be little r and little y. So remember, these are our gametes. So let's go ahead and go with say that's the egg, and this one's the sperm. These are our gametes. And then they are going to combine in fertilization to now come up with the offspring that has the genotype big R, little r, big Y, little y. And what's the phenotype of this individual right here? Well, remember, the round is dominant, so the seed's going to be round, and yellow is dominant. So the seed's going to be yellow. Okay, so that is our first generation right there. That's what we're looking at. So our phenotype, round yellow seeds, our genotype, big R, little r, big R, little r, big Y, little y, big Y, little y. So that is our first generation. Okay, so now we are going to cross that first generation, and let's see what we get. Okay. And we'll do that in the next video. Okay.